This conference will now be recorded.
Hello students. Now we are learning about plant growth and development, chapter number 15. So in this chapter you have to learn about the plant growth and development. So you need to start with seed germination and phases of plant growth and plant growth rate, then conditions of growth, differentiation, de-differentiation and redifferentiation, then sequence of developmental process in plant, growth regulators like auxin, gibberellin, cytokinin, ethylene and abscisic acid. About all these things we are learning. So we have already covered what is plant growth and types of growth curves we have learned, and what is growth rate. In that, arithmetic growth and geometrical growth both we have learned. Then the conditions of growth, like how the nutrients, what are all the essential requirements of growth in plants, like water, nutrients, temperature, oxygen, light and gravity, how these things are affecting the plant growth. Then what is differentiation, what is de-differentiation and what is redifferentiation. About all these things we had discussed. And the sequence of development process in plant cells, that sequence, like from meristematic cells, it is dividing, growth is happening there, and new cell, then the expansion, then the enlargement, maturation, then a mature cell, it will grow and it will be there for a certain stage, and then senescence, which is leading to death of that. So the sequence of developmental process in a plant cell also we have learned. Then the plant growth regulators. About plant growth regulators we need to learn, like there some plant growth promoters and growth inhibitors are there. And before that, in the previous class we have learned about seed dormancy, then seed germination, all those things also we have learned. So in this class now we are going to learn about the plant growth regulators. Okay, so plant growth regulators. So here in this chapter, now in these classes, 10 previous year question papers, previous year question papers from the last 10 years question papers. I'm collecting all the questions, and along with that some extra topics also adding, and like that I'm handling the classes. So here, if you are learning these topics, it's equal to last previous year question papers, like previous 10 years question paper you are covering. So in this way, after learning all these topics, if you're checking your previous year question papers, it will be easy for you to answer. So here now we are going to the remaining topics of this chapter, plant growth and development. So first we are starting with plant growth regulators. Plant growth regulators, or we can write PGRs, plant growth regulators. Clear? Here, plant growth regulators or phytohormones. Plant growth regulators we can call as phytohormones. Okay, so these phytohormones or plant growth regulators, they are small and simple organic molecules. Okay, they are organic molecules, and they are very small molecules, and they have diverse chemical composition. Okay, they have different chemical composition. They have different chemical composition, and all these plant growth regulators, they are naturally produced in plants. Okay, they have already present in plants, or they are naturally getting produced in plants. We are not introducing into plants, they are naturally getting produced in plants. And these plant growth regulators, see from the name itself it's clear, plant growth regulators, it is regulating the stages of growth stages. In different stages it is getting uh, the function, or it is getting uh, it is trying to regulate the growth of plants. So here in plants it is regulating one or more physiological functions. Okay, it's regulating the physiological functions. Clear? So that is trying to regulate the physiological functions and different stages. And these plant growth regulators, they are divided into groups. What is that? First one is growth promoters. Growth promoters. And the second type is growth inhibitors. Growth inhibitors. Okay, so growth promoters and growth inhibitors. In that first one, growth promoters. What is growth promoters? Let's see. Growth promoters means they are the chemical substances or the phytohormones which perform the growth promoting activities, that promotes the growth. It is doing the activities which is promoting the growth, like cell enlargement, cell division, seed formation, flowering, fruiting. All these are showing the growth stages, right? Different growth stages. So this is promoting the activities like cell division, seed formation, flowering, fruiting, like growing stages. It is moving from one stage to other stage, so it is showing the growth. So these are the chemicals which is promoting the, which is showing the performance, which is performing like growth promoting substances. Okay, and what are growth inhibitors? Second type is growth inhibitors, we said. Growth inhibitors means those things are the chemical substances which is performing growth inhibiting activities. These chemical substances will show the inhibiting activities of growth. Okay, like we can say the dormancy, abscission, all these things. Okay, so growth promoters, example we can say auxin, cytokinin, gibberellins, all these are growth promoters. Auxin is growth promoter. Cytokinin, that is also growth promoter. Then gibberellins, this is also growth promoter. These are the examples of growth promoters. And growth inhibitors means they are showing the inhibiting activities, right? So abscisic acid, ethylene. Example, abscisic acid and ethylene. These are the growth inhibitors. Okay, now we need to learn different hormones and their location, physiological effects, and the uses we need to learn. Okay, so we'll learn one by one. First is we will learn about the hormone auxin. Auxins. See, this auxins, these are indole derivatives. Okay, auxins, they are the indole derivatives, and these are first isolated each and every point of this, like their hormones, location, physiological effects, and uses. Each and every points which I'm saying, these are from previous year question papers. Okay, so you need to learn all these points without missing one point. You need to learn. Okay, so the auxin, which is indole derivatives, then these are first isolated from human urine. First isolated from human urine. Which one? Auxins. And its name, it is given by F.W. Wendt. Okay. And the existence of growth substances, that was first proposed by Charles Darwin and his son Francis Darwin. So growth substances existence, like hormones like this, the existence of these kinds of hormones, that was first proposed by Charles Darwin and his son Francis Darwin. This question one time only came. Then next, the important point, the location and the physiological effects we need to learn. This auxins that is located at apical meristems and other immature parts of plants. Apical meristem, see the growing regions, these are growth promoters, right? Growth promoters, auxin is a growth promoter. So this is located at apical meristems and the other immature parts. Other immature parts of plants. Okay, so auxin that is located at apical meristem and other immature parts of plants. And what is the function, physiological effects, what change it is making? This is helping for cell enlargement, then this initiates and enhances cell division, helps for cell division, initiation and enhancement of cell division, for that also it is helping. And apical dominance, that is due to the presence of auxin in apical buds. That is, apical dominance is shown due to the presence of auxin in the apical buds, and this inhibits the lateral buds to grow. So the apical dominance, that is due to the presence of which one? Auxin, which is present in the apical buds, and these apical buds or the presence of this hormone auxin at the apical side, that inhibits the lateral buds to grow. That creates a situation for the inhibition of the lateral bud formation. Clear? And this auxin that causes root initiation. And see, this is growth promoter. So this delays the abscission of leaves. Abscission of leaf, abscission of root, for all these things, this delays. Okay. Next comes so these are the points about the location and the physiological effects what it is creating, like apical dominance, root initiation it is helping, and that delays the abscission of leaves and fruits. So all these, and it is helping for cell enlargement, help for cell division, all these are the physiological effects. And the location where it is present, it is located at the apical meristem and other immature parts of plants, okay, in the growing region. Next comes the uses of auxins. So let's see what are all the uses. In parthenocarpy. In parthenocarpy, which and all was IAA, IPA, NAA, then IBA. Okay, so these are indole derivatives. Indole derivatives we are saying. So here IAA, IPA, NAA, and IBA. These are the indole derivatives. Next one, this is work, it is using for parthenocarpy and the effective weedicide. It is working as effective weedicide for killing weeds, or in that it is working like 2,4-D, NAA, all these are effective weedicides. All these are working as effective weedicide also. Then flowering, it's a growth promoter, right? So in the flowering also it is working NAA and 2,4-D. This is helping for flowering also. Then again for cell division it is helping. So callus and cell suspension culture, in that also it is working. Callus and cell suspension culture. Then auxin, when it is coming in combination with the cytokinin, that is used in the tissue culture for inducing morphogenesis in the callus. So auxin plus cytokinin in a particular ratio, when it is combining and using, this is used in the tissue culture for inducing morphogenesis in callus. In tissue culture side, it is useful. Okay, so these are the uses of auxin. Next comes the bioassay. So what is this bioassay? Bioassay means testing of biological activity. That means the growth or the biological activity of a substance. Here the substance is auxin. Okay, by giving some living material, that is called as, or by employing some living material, that is called as bioassay. So testing of biological activity. We are testing the biological activities of auxin here by supplying auxin um, for some living material. So what are all activities it is showing when we are giving the auxin, and that auxin how we will give in different dose, we will give in different ways, we will give at different stages, we will give, so that we can make out which and all, what and all changes it is making. Okay, so such type of work, that is called as bioassay. So from here, bioassay, one question came, and another one in avena coleoptile. In that curvature test, this was carried out by F.W. Wendt. This is one question. Then he demonstrated the effect of auxin on plant growth by performing experiments with the oat coleoptile. Effect of auxin on plant growth by using oat coleoptile. So Avena coleoptile curvature test was done by F.W. Wendt in 1928, and he demonstrated the effect of auxin on plant growth by performing experiment with which one, which oat coleoptile. So these both questions came, these both points are important and these are repeated. Next question, split pea stem curvature test. Split P-stem curvature test. This was also discovered by Wendt in 1934. Okay, split P-stem curvature test. That was also by Wendt in 1934. Then root growth inhibition test. These are the bioassays done for auxin. Root growth inhibition. All these are coming under the bioassay of auxin. Okay, so all these points you must learn without leaving one point, and from this 2-3 questions are repeated. Okay, next we are moving to gibberellins. Gibberellins. Gibberellins, these were discovered in Japan in connection with Beckley disease or foolish seedling. Okay, so this was discovered in Japan. So it is connected with the Beckley disease or foolish seedling of rice, which is caused by the fungus Gibberella fujikuri. Okay, and Sawada discovered that some substances which is secreted by this fungus, which one? Gibberella fujikuri, the fungus, that is responsible for the disease. See, we said Beckley disease or the foolish seedling of rice, which is caused by the fungus Gibberella fujikuri, and that fungus is secreting some substances. That substance which is secreted by the fungus is responsible for that disease. Okay, and Kurosawa induced Beckley symptoms in healthy rice seedlings by applying the extracts of this fungus. Okay, Kurosawa induced this Beckley disease symptoms. Okay, in a healthy seedling. In a healthy seedling, they introduced these Beckley symptoms in a healthy seedling. And how they induce these symptoms? By applying the extracts from these fungus that were given to a healthy seedling to induce the symptoms of this disease. And Yabuta and Samuki, they isolated and crystallized the substance and gave this name gibberellic acid. Beckley disease symptoms in healthy seedling of rice by applying the extracts of this fungus. Okay, then Yabuta and Samuki, they isolated and crystallized the substance. Isolated and crystallized this substance and gave the name. They gave the name which one? Gibberellic acid. Okay. So here 2-3 scientists name it's coming. So these are important. And next we are moving to the location and the physiological effects. Location and the physiological effects of gibberellic acid or gibberellin. So here this is located at the roots and shoot tips. Where and all you can see root tip, shoot tip, young leaves, then seeds. In plant seeds you can see which one, this gibberellin. Then this creates some situation like initiation of flowering. Initiation of flowering in which kind of plants? In vernalized and long day plants. In vernalized and long day plants, gibberellins, that creates a situation or the initiation of flowering during short day condition. In long day plants, during short day conditions, this is creating the initiation of flowering. For example, we can say in cabbage. Then another thing is this is promoting the bolting. Bolting means the internode elongation. Just before flowering, that internode elongation will come. So that promotes the bolting. Then in beet, in cabbage, and in many plants with rosette habit. So we can give promote bolting. Okay. The next point is this stimulates the production of hydrolytic enzymes like amylase, lipases, proteases. So this mobilize the stored food during seed germination. Okay, so stimulates the production. Which one? This gibberellins that stimulates the production of hydrolytic enzymes. Hydrolytic enzymes, which are all the hydrolytic enzymes like it is stimulating lipases, amylases, proteases, amylases, proteases, all these. So that is mobilizing. That's how it is mobilizing the stored food during the seed germination period. Okay. And next point, under this, the location and physiological effects, we are learning the last point, spraying that juvenile coniferous. Okay. So spraying juvenile conifers with gibberellins or gibberellic acid that hasten the maturity period, and that is leading to early seed production. So in the juvenile conifers, if you are spraying this gibberellic acid, that makes the fastening or it makes it fast maturity period, and that's how it is doing the early seed production. Okay, in juvenile conifers, uh, spraying of gibberellic acids or gibberellins that hastens the maturity or that makes the maturity period very fast, and that's how it is moving to seed production. So that is the role. Which one is doing that role? Gibberellins. Okay, so here the physiological effects, the physiological effect which is created by the gibberellin that we can see here, it is when we are spraying this gibberellic acid in juvenile conifers, there we can see an early maturing and the seed production. Clear? So now we have learned the physiological effects, and next is the uses of gibberellins, that we need to say that enhances the size of seedless grapefruits. Size of seedless seedless grape, now it's commonly available. So that gibberellins or this gibberellic acid that enhances the size of that seedless grapefruit and enhances the height of sugarcane plants. In sugarcane plant, it increases the height or enhances the height, and if it is increasing the height, what will happen? Yield that will increase, that increases the yield of sugar. Then again, it is used in the production of seedless pomaceous fruits. Production of seedless pomaceous fruits. Okay, so for the production of seedless pomaceous fruits, that is also for that also this gibberellic acid or gibberellins are used. Okay, and when we are moving to the bioassay of gibberellins, there we can see this gibberellins that is performed through dwarf maize or P test and serial endosperm test. So bioassay of gibberellin, there we can mention 3 tests like dwarf maize, P test, serial endosperm test. So these are the bioassays of gibberellin. Okay, so here gibberellins, another important point, gibberellins, they are the terpene derivatives. This question, terpene derivatives, gibberellins, they are the terpene derivatives, and this question came 3 times. Okay, so here first we need to learn gibberellins, what are they? They are the terpene derivatives. Then you should move to the discovery. So here the disease, Bacony disease or foolish seedling of rice, which is caused by the Gibberella fujicore, and that was discovered in Japan, right? And along with that, the discovery of this gibberellins happened. Okay, so this Sawada, Kurosawa, Yabuta, and Samuki, all these scientists' name along with their role in this, in gibberellins, that you need to learn. Then where it is located, and that stimulates the production of these enzymes. And in juvenile conifers, if you are spraying gibberellins, what will happen? That point you need to learn. Then what are all the uses? So size of seedless grape enhances the height of sugarcane plant, that is also increasing, and that's how the yield also increasing. Then in seedless pomaceous fruits, that also can be produced. Then in which and all bioassay these things are getting used. Okay, these are the main points which we need to learn. And next is the other hormone, cytokinin. Cytokinins. Here, cytokinins, these are adenine derivatives. Adenine derivatives. Which one? Cytokinins. Okay. And here, discovery, Skoog and Miller discovered a substance, kinetin. Okay. Skoog and Miller. They discovered a substance, kinetin. From where? From DNA. Okay. Then Latham and Miller, they isolated a cell division inducing substance from the milky stage grain of maize, and they named it as zeatin. That was the first natural cytokinin. Who discovered? Latham and Miller. Latham and Miller. From milky stage grain of maize. Okay, so milky stage grain of maize. From that, Latham and Miller isolated a substance which is inducing cell division. That substance was inducing cell division. Okay, and they named the substance as zeatin. Okay, and this was the first natural cytokinin. First natural cytokinin. Which one? Zeatin. And this kinetin, we said the cytokinin, the kinetin, this induces cytokinesis, and that's why it is named as cytokinin. So kinetin, this kinetin substance induces cytokinesis. Cytokinesis or cell division, that's why it is named as cytokinin. That's why the name is given as cytokinin. Then the location and the physiological effects of this cytokinin we need to learn. Location and physiological effects that we need to learn of cytokinin. See, the cytokinin, this is located at the root apical meristem and in immature fruits. Okay, in root apical meristem and in immature fruit it is present, and this we said this induces cell division and cell expansion. Cell division and expansion of cell. For that it helps, and that helps or that promotes the organogenesis when it is used along with auxin. Along with auxin, that promotes organogenesis. Okay, and this cytokinin that delay the senescence or the yellowing of leaf. Yellowing of leaf or senescence, it is delaying, and that's called as Richmond-Lang effect. Okay, and this is helping to overcome the apical dominance. For helping the apical, for helping the to overcome the apical dominance, that is helping. Okay, and stomatal opening, this cytokinin that promotes the stomatal opening. Another question which came, this increases, cytokinin increases the shelf life of vegetables and flowers. Increases shelf life of vegetables and flowers. Which one? Cytokinin that increases the shelf life of vegetables and flowers. So all these questions came in previous year question paper. So this is located at the root apical meristem and in immature fruits, and that promotes organogenesis when, along with oxygen. Okay, not only this one, along with oxygen it is helping for organogenesis, and for cell division and cell expansion it is having a role, and the delays in senescence or yellowing of leaf, that is called as Richmond-Lang effect, and that promotes stomatal opening, increases the shelf life of flowers and vegetables, and it is helping to overcome the apical dominance. All these are the physiological effects coming under the physiological effects, and where it is located? Located at root apical meristem and in immature fruits it is located. So that's the location. Next comes about the uses. Here uses when we are saying the promotion of chloroplast development. In the chloroplast development, there we can say, and the promotion of bud formation. Bud formation promotion. In all these are the uses: promotion of chloroplast development and bud formation. Okay. Then the bioassays. Here, bioassay that is done through the retention of chlorophyll by leaf disc and gain of weight of a tissue in the culture, then excised radish cotyledon expansion and root inhibition test. These are the bioassays. Bioassays done through this retention of chlorophyll by leaf disc, then expansion, this cotyledon, excised radish cotyledon expansion. The excised radish cotyledon, in that also bioassay is done, the expansion and root inhibition test. Okay, then all this gain of weight in a tissue in the culture. For all these bioassays, which one is used? Cytokinin. So these are the points which you must learn about cytokinin. Next comes the ethylene. So now we have learned about auxin, gibberellin, and cytokinin, which are the growth promoters. Okay. Next, about growth inhibitors, which are the substances which performs the inhibiting activities. That about those we are going to learn. So first we will learn ethylene, which is a growth inhibitor. So this is derived from methionine. Derived from methionine. Okay. This methionine, first about discovery, when we are saying Croker reported the presence of ethylene from plant organs, and he named that as gaseous hormone. Gaseous hormone. Which one? Ethylene. That is the gaseous hormone which is reported. The presence of this ethylene is reported from plant organs, and Croker named it as gaseous hormone. And where it is located? This ethylene is located at root, shoot, apical meristem, leaf node, and ripened fruits. Everywhere it is located. Roots, shoots, apical meristems, leaf nodes, ripened fruits. And this ethylene, this is highly effective in fruit ripening. In fruit ripening, this is having a very good role. And ethylene, this promotes abscission and senescence of leaf and flower. Then that promotes the root growth, root hair formation, root growth it promotes, then root hair formation it promotes. Okay, when root hairs are more, what will happen? Due to the more root hairs, the plant increases the ability to absorb more, right? Because the absorption surface is increasing when it is getting more root hairs. So through root absorption will be more. Then germination in peanut seeds and sprouting of potato tuber, that is promoted by ethylene. Peanut seed, ethylene promotes germination in peanut seed. Then sprouting of potato tubers. Okay. Then another one important point, flowering and synchronizing fruit which is set in pineapples. Fruit set in pineapple, for that flowering and synchronizing, that also ethylene can promote. Clear? So all these are the questions which is coming for NEET exam. It's already came in the previous year question paper. So this may repeat because this kind of questions you will get confused about which hormone is this one. So here ethylene and abscisic acid, which are the growth inhibitors, and the other cytokinin, gibberellin, auxin, all these are growth promoters. So now about ethylene we are learning about the location and physiological effects which is created by ethylene, which is a gaseous hormone. That's what we have learned. Next about the uses. So the ethylene that is most widely used compound which is the source as a source of ethylene in agriculture, that is ethipone. This is the agriculture source. So this here you can say that that makes the fruit ripening fast in tomato and in apples, and that what is it is doing, ripening, it is having a major role in the ripening of fruits, right, in the tomato, then apple and all, then that accelerates the abscission in flower and fruit. Okay, these are the major uses. Then about bioassay, when we are saying about the bioassay of ethylene causes triple response in plants. Triple response in plants. Okay, that means thickening of stem and nodes, then inhibition of elongation of internodes of stem, then the induction of horizontal growth of stem against the gravity. These are the triple response. So bioassay of ethylene, for that triple response in plants. Okay, next hormone comes which is the growth inhibitor, that is abscisic acid or ABA. Okay, this is stress hormone, and this is derivative of carotenoid. Derivative of carotenoid. Okay, so this hormone, this was first isolated by Edicott et al. Edicott et al. Then from cotton fruits, this was first isolated and that was named as abscission. Okay, and simultaneously Conforth and Waring, they isolated the substance which can induce bird dormancy, and they named the substance as dormine. So later, both these substances were found. Uh, these are the same substances like that they found, and the same were named as both of them. Like both the substances are same. Later they proved it, and they both named them as abscisic acid. Okay, and where you can see the locate, this is located, abscisic acid is located in leaves, fruits, root cap and seeds. Okay, and that induces seed and bird dormancy, induces seed dormancy and bird dormancy. seed dormancy and bud dormancy, it is induces. That's why we said these are growth inhibitors. So it is inducing such characters, and that promotes the closing of stomata. Closing of stomata it promotes, and when it is doing that in water stressed condition, so there it will reduce the decrease in transpiration rate. It is making less by closing the stomata in a water stressed condition, right? So that's how it is helping. So here that promotes the senescence. Which one? Abscisic acid promotes the senescence. That means shedding of leaves, fruits, or fruits, that promotes. And in long day plants, that inhibits the flowering. Inhibition of flowering in long day plants. Okay, and cell division and cell elongation, that is delayed. Here these are growth inhibitors. It delays the cell division and cell elongation, abscisic acid. And this uses of this abscisic acid, we can say this is naturally occurring growth inhibitor in plants, and that can act as the enemy of this gibberellin, or it can work against, like it can act as antagonist to what? Gibberellins, right? It is a growth promoter. Here this one is against growth promoting activities. And next comes the, so this is a naturally occurring growth inhibitor. Which one? Abscisic acid. So it will work against the, which one? Gibberellins, which is promoting the growth. Next, about the bioassay of abscisic acid. So if you are learning about this growth hormones which is promoting or inducing, whatever it may be, you should remember the derivative. It is the derivative of which one? Then about the discovery, then the location and the physiological effects, then the uses and bioassays. These are the 5-6 points which you must learn about each hormone. Okay, then last one which we are learning about abscisic acid, that is a bioassay of abscisic acid and rice seedling growth inhibition test. In that, they have used an inhibition of alpha amylase synthesis in barley. So rice seedling growth inhibition. Okay, in rice seedling growth inhibition test, they have used and the inhibition of alpha amylase in the alpha amylase synthesis in barley. So in both inhibition test, bioassay is done for abscisic acid. Clear? So all the hormones you have completed, and you must learn these 5 to 6 points about each hormone. So next, the last topic which we are learning in this chapter, that is photoperiodism and vernalisation also we need to learn. Photoperiodism first. See, photoperiodism, that is the effect or requirement of relative length of day and night for flowering. Relative length of day and night. Photo means light, periodism means that time length. Okay, so that relative length of day and night which is required for flowering. So that is the what effect it is creating. So this is the effect or the requirement of the length of day and night. Relative length, we can say day length is more, so like that. So photoperiodism was first studied by Garner and Allard. Garner and Allard first studied this photoperiodism, and in which one? In Maryland mammoth. It's a tobacco variety. It's a tobacco variety. Okay. And every plant has a specific critical period of light duration which is needed for flowering. Every plant is having. Okay. So we can classify the plants, short day plants, long day plants, day neutral plants, short long day plants, long short day plants. Like that, we can classify the plants. Okay. The next topic is vernalization. So this vernalization, we can say about vernalization, like Russian agronomist T.D. Lysenko. He coined this term vernalization. So the process of conversion of winter variety into spring variety. Winter variety is getting converted into spring variety by providing low temperature or chilling treatment. That is called as vernalizations. So we are converting the winter variety into a spring variety. How? By providing low temperature. Providing low temperature or the chilling treatment we are providing, and we are converting the winter variety to a spring variety. So that is called as vernalization. So this process of vernalization that stimulates, what it is doing, that stimulates the stimulus, which is how we can explain the process means, in the process, in the vernalization process, the stimulus which is received by the cells, stimulus will go to cells, and that cells, the stimulus which is gone to that cells, that will travel to all the parts of plant, that will go to all parts of plants, right? And that prepares that plant to flower. Okay. So here, vernalization, how we can summarize, that is a process of shortening of vegetative phase. There the vegetative phase is getting shortened, and that is hastening, or that is making fast the flowering period by cold treatment. Okay, so that can be devernalized. How it can be devernalized? By exposing the plant to temperature of 30 degrees centigrade or above. That's how we can devernalize. So vernalization, we can say we are shortening the vegetative phase and trying to bring fast the flowering and flowering of that plant. How? By cold treatment. Okay. So how devernalization is like that. So devernalization means exposing that plant to the temperature of 30 degree or above. That is the devernalization process. And how this vernalization is doing? The site of vernalization, that is meristematic cell. In meristematic cell, there the vernalization process is doing. So that produces these meristematic cells produces a hormone vernalin. That hormone, which is that hormone? Vernalin hormone is getting produced. Um, that thought to be gibberellins or the mixture of gibberellins, and this is involved in the initiation of vernalization stimulus. Clear? So that's all about the vernalization. So now we have covered almost all the topics of this chapter, which is came in the previous year question paper. So topic 1 you need to learn, that is plant growth and plant hormones, and topic 2 is photoperiodism, vernalization and senescence. Then all the hormones, um, all the points, almost all the points from the previous year question paper we have covered. So go through all these topics and prepare well. So in the next class we will do another chapter. Thank you.